Hi, and welcome to Chapter 5. In this chapter, we're going to talk about the eukaryotic cell and the microorganisms that are eukaryotes that we study in microbiology. And the article at the beginning of your Chapter 5 is about the use of the neti pot. The problem with this is some people were improperly using water from different sources in these neti pots, and the problem became that some of that water was contaminated with this guy here. And this is actually the three different stages of Naegleria fowleri, more commonly known as the brain-eating amoeba. It is a eukaryotic cell found in most fresh waters that is untreated, and what we found was there was a few cases that people were using neti pots that had the Naegleria fowleri in it, and this actually got up through their cribriform plates in the base of the skull and infected their brains, and actually they died from this organism. So let's first talk about the history of the eukaryotes. Eukaryotic cells first came on the scene some 2 to 3 billion years ago. And they all actually came from the last universal common ancestor. The LUCA was neither a prokaryote or eukaryote, but simply the ancestor of all cells. This gave rise to the bacteria, the archaea, and the eukarya all separately. So they didn't actually develop together, but they actually developed separately from each other. And one of the big things that separates eukaryotes from the other prokaryotes is the formation of organelles. These originated from more primitive cells that became trapped in the eukaryotic cell or were actually indentations of the cell membrane themselves. So that brings us to the endosymbiotic theory that was first proposed by Dr. Lynn Margulis. Her theory states that as the ancestral cell began to grow, it actually invaginated the cell membrane to form the nuclear membrane around the single circular chromosome. And then that the single circular chromosome was adding to its mass and was actually then chopped up into the linear chromosomes that we now know in eukaryotic cells. Also, things like the endoplasmic reticulum and Golgi apparatus were formed by the same invagination of the cell membrane. But as the cell continued to get larger and larger, it needed more energy production than it was able to do by itself. So what the endosymbiotic theory states is that this larger cell then began to incorporate prokaryotic cells that then became the mitochondria and the chloroplast. And that actually makes sense in the fact that today's mitochondria and chloroplasts do contain their own circular DNA, and they do contain ribosomes very similar to the 70S ribosomes that we see in the prokaryotic cells of bacteria. So this may actually be the theory that it's where these actually developed from, is that this last universal ancestral cell began to grow and branch off into the eukaryotic cells. The first primitive eukaryotic cells were single cell and independent from each other. They became specialized to perform a particular function within colonies, and this complex then multicellular organisms evolved when cells lost the ability to survive apart from the colony. And this then led to the formation of the tissues, the organs, and finally the organ systems. So eukaryotic organisms that are studied in the field of microbiology include the protozoa, which are always unicellular and most of the time are found in water, the fungi and algae, which may be unicellular or multicellular, and the helminths, which includes the worms and the flukes, which are always multicellular, but they have unicellular eggs or larvae forms. And these eggs and larvae forms are oftentimes microscopic. So, true or false, eukaryotes and prokaryotes evolved independently of each other. What evolved from the primitive cells that became trapped in larger cells that we now know today as some of the organelles? Describe the differences between tissues and organs. And list 3 types of eukaryotes that we study today in microbiology. So, features of eukaryotic cells, and this should actually be some review from anatomy and physiology. Things that are found in all eukaryotic cells, of course, are the cytoplasmic membrane, the nucleus, the mitochondria, endoplasmic reticulum, Golgi apparatus, vacuoles and vesicles, cytoskeleton, and the glycocalyx, which is oftentimes referred to as the matrix in eukaryotic tissues. Found in some eukaryotic cells will be a cell wall made up of primarily sugars, locomotive appendages such as flagella or cilia, chloroplasts in those photosynthetic cells, and this is a picture of the generalized eukaryotic cell. So remind yourself of all of these, and we're going to go quickly through some of these organelles and their primary functions. So let's start from the outside inward, the cilia and flagella. The eukaryotic versus bacterial flagella is one that the eukaryotic flagella is much thicker, almost 10 times thicker. It is structurally more complex. It is covered by an extension of the cell membrane itself. And there's long sheathed cylinder proteins that contain regularly spaced hollow microtubules in what we call the 9-2 arrangement of the microtubules. And so this is what the base of the eukaryotic flagella actually looks like. It doesn't have the hook that we see in the prokaryotic flagella, so this does not spin. It actually undulates back and forth. And this is what we call the 9-2 arrangement. So this is a cross section of a flagellum. We have 1, 2, 3, 4, 5, 6, 7, 8, 9 of these doublets around the outside of proteins, and we have a center doublet in the middle. And this allows for that undulating movement of the flagellum. The cilia are similar in overall structure to the flagella, but they are shorter and more numerous, up to several thousand in some cells. They're found only in a single group of protozoa and certain animal cells. They function as both feeding and filtering structures on some cells, and they're involved, of course, in movement. And they have directional movement. They first have what's known as the power stroke, and this is actually what will drive the cell forward in the environment or move stuff along the surface of the cell that has the cilia. And then they go into a recovery stroke, and they simply then flip back to their original position to then redo the power stroke again. There is no movement in the recovery stroke, and so this will allow directional movement of either the cell itself or the stuff on the surface of the cell. And this is our good old paramecium. And here we have the cilia all along the outside of this protozoan parasite, and these are actually involved in locomotion of this organism through its water environment. The glycocalyx, or the matrix. This is an outermost boundary that comes in direct contact with the environment. It's composed primarily of polysaccharides with some proteins thrown in. The appearance, it typically has almost a fibrous-like network appearance, and it's somewhat similar to the slime layer that we saw in the bacteria in the fact that it is not compressed highly. A few of the eukaryotic cells form almost a capsule-like structure as well of the glycocalyx. The cell wall, in those eukaryotes that have cell walls, they are found in the fungi and the algae. It's rigid and provides structural support and shape, just like we saw in the cell wall of the bacteria, but they're different chemical composition than the peptidoglycan in bacteria. Primarily, we find chitin or cellulose in fungi, and we certainly saw cellulose in plant cell walls. So this actually represents the cell wall of a yeast body here. And so we have the glycocalyx, we have the mixed glycans, glycoproteins, and then actually chitin that forms this cell wall of this particular yeast. The cytoplasmic membrane is again that typical layer of phospholipids embedded with protein molecules, but in eukaryotic cells we have many sterols, such as cholesterol, that kind of holds together the phospholipid bilayer. This will stabilize the membrane. This membrane is highly selectively permeable and has sophisticated mechanisms for transporting materials in and waste and other products. So just a snapshot of the bilayer of phospholipids. Here's some cholesterol thrown in. I kind of like to think of cholesterol as the mayonnaise that holds the two pieces of bread together. And then we have these proteins and glycolipids and glycoproteins that float around in this fatty lipid membrane. So describe 3 ways in which eukaryotic flagella are different than prokaryotic flagella. What's the purpose of cilia? What are the 3 functions of the glycocalyx? And describe 3 ways in which eukaryotic cell walls are different than prokaryotic cell walls. The nucleus. This is the control center of the eukaryotic cell. Compact sphere is the most prominent organelle that we see in the cell. It's separated from the cytoplasm by the nuclear envelope or nuclear membrane. There are 2 things contained within the nucleus. The nucleolus, which is actually a structure of RNA, and it tends to stain more intensely than the other component due to its RNA content. The nucleolus is the site of ribosomal RNA synthesis. This is where we make the subunits of the ribosome. And this actually shows the nucleolus. So a darker staining body than the rest of the content. And there's the nuclear membrane. You will note that there are nuclear pores in the membrane, and this allows for the passage of large structures primarily out of the nucleus. The other part contained within the nucleus is the chromatin. Now, the chromatin is DNA. Chromosomes are DNA. It's simply how tightly are they packaged. Chromatin actually is the loose arrangement of the DNA within the eukaryotic cell, while chromosomes are tightly packed around a group of proteins that will compact them for cellular division. And so chromatin contains the unit of genetic information, or the genes, within the cell. They're long linear DNA molecules, and they are multiples, and they are diploid in number in the normal cell. They are bound to a group of proteins called the histone proteins, and this will allow for them to be either loose or compact in nature. They are only visible as chromosomes during mitosis and meiosis. Mitosis is the division of the somatic or body cells, while meiosis or meiosis is the production of the sex cells. And just a review of mitosis. So we have a copy of the chromosome made, and they are held together at the centromere. And so we have the 2 sister chromatids that give us this appearance of the X shape of the chromosome. So here are the chromosomes. The nuclear membrane will dissolve during the first stages of mitosis. The centrioles will actually then develop the spindle fibers. So the centrioles will move apart from each other, develop the spindle fibers. The chromosomes will align and eventually align right along the equator of the cell during mitosis. And then the 2 sister chromatids will split at the centromere and be drawn by the spindle fibers towards the poles of the cell by the centrioles and spindle fibers. And this is anaphase. And then the cell actually undergoes cleavage, which will then separate the 2 now reforming nuclei in telophase into 2 separate daughter cells that have the exact same number as the original parent cell. So review your stages of mitosis. The endoplasmic reticulum are a series of membrane-bound channels that run through the cytoplasm that are used in transport and storage of different substances. There's 2 forms: the rough endoplasmic reticulum that has ribosomes attached to its outer surface, and this is where proteins will be made and then held in the ER for packaging and transport. The smooth endoplasmic reticulum has no associated ribosomes and primarily function in synthesis and storage of non-protein molecules. So here's an example of the rough endoplasmic reticulum with the associated ribosomes on the outside of the double membrane. The Golgi apparatus is also a double membrane bound organelle, and it's the site of protein modification and transport. Consists usually of 6 to 9 or so stacks of flattened disc-shaped sacs, usually closely connected to the endoplasmic reticulum. Where it is close to the Golgi apparatus, the ER will form transitional vesicles that pinch off from the ER membrane that then are picked up by the Golgi apparatus. The Golgi apparatus then will modify the proteins, adding maybe polysaccharides or lipids to the proteins, or modifying the protein chain of amino acids. And then these are pinched off into condensing vesicles that then are conveyed to other organelles or transported to the cell membrane as secretory vesicles. So this just shows the endoplasmic reticulum. It pinches off with protein in these transitional vesicles. They then bond to the cell membrane or the membrane of the Golgi apparatus, where it's then modified, and then we can pinch them off in these condensing vesicles that then will go to other organelles and deliver their product or to the cell membrane. And this is just the Golgi apparatus here on this photomicrograph. So, genetic information originates from the nucleus, and this is where we have the blueprints to make proteins, and we'll talk about protein synthesis a little later on. Proteins are synthesized at the ribosomes and will be deposited into the rough endoplasmic reticulum that are then transported to the Golgi apparatus and are chemically modified and packaged into vesicles. So we really have an assembly line. So we read the blueprint, we then send out that information to the ribosomes make the protein that's stored in the endoplasmic reticulum, we pinch off the transitional vesicles, send it to the Golgi apparatus, the protein is modified, and then will go out for delivery. Cellular vesicles can contain several different things, and oftentimes just looking at a picture, I can't tell you exactly what the vesicle is until I know what it actually contains. And one of the most numerous of the cellular vesicles is the lysosomes. These originate in the Golgi apparatus and contain a variety of digestive enzymes. The lysosomes are primarily involved in intracellular digestion of food, and the digestion of invading microorganisms. Vacuoles are simply membrane-bound storage sacs, and these can actually store a variety of products. It can contain fluids or solid particles that are to be digested, excreted, or stored within the cytoplasm. So if I were just to look at these in the picture, I couldn't tell you, is it a lysosome? Is it a vesicle vacuole? Is it a peroxisome? I don't know. What I need to know is what is inside it. If it's just containing digestive enzymes, it's a lysosome. If it is a lysosome that's actually fused with a what we call phagosome, a vacuole that's been endositized, we then refer to this as a phagolysosome. And those digestive enzymes will digest whatever is inside of this phagolysosome. The mitochondria, the powerhouse of the cell. This supplies the bulk of energy in all eukaryotic cells. It is a double membrane bound organelle, meaning that it has both an inner and outer membrane. The inner membrane is folded to form shelf-like structures called cristae, and it is on these cristae that hold the electron carriers of aerobic respiration for the production of ATP. The matrix, which is the fluid inside the mitochondria, will hold ribosomes, which are 70S ribosomes, DNA, that are small circular pieces of DNA, and other enzymes used in metabolism. The DNA, the genes on this DNA of mitochondrial DNA, are genes involved in the production of enzymes for metabolism. Mitochondria can divide by binary fission independently of the cell that it is in. It contains those small circular strands of DNA, and it has bacterial size 70S ribosomes. The ribosomes on the ER floating in the cytoplasm of the cell are 80S ribosomes. So these characteristics provide evidence that the mitochondria were probably small prokaryotic cells that were engulfed by the growing eukaryotic cell and became these organelles in the endosymbiotic theory. And this is just a picture of the mitochondria. So here's that inner membrane folded into these shelf-like structures of the cristae. The matrix is the fluid. Chloroplasts, in those photosynthetic eukaryotic cells, these are what are capable of converting energy of sunlight into chemical energy through the process of photosynthesis. And we'll talk about this in the metabolism chapter. Photosynthetic role of chloroplasts, this is the primary producer of all organic nutrients. Really, photosynthesis was first. It was the first thing that came along before we had the other forms of metabolism. And it is the photosynthetic organisms are the primary producers of oxygen gas for the atmosphere. This is just showing the structure of a chloroplast in a eukaryotic cell. We have stacks of these, what we call thylakoids, in what's known as the granum, which is the stack of these thylakoids. And this actually winds up forming a double membrane, similar to the mitochondria. So we have an outer membrane and the inner membrane of the thylakoid. And this is where we're going to see that chemical energy produced from the light energy of the sun. Chloroplasts also have 70S ribosomes and their own circular DNA. So ribosomes. All ribosomes, whether they be 70S or 80S, are protein synthesizers. Ribosomes will be distributed throughout the eukaryotic cell. They can be free-floating in the cytoplasm and located along the cytoskeleton microfibrils and tubules, or they can be attached to the endoplasmic reticulum. They are also found inside the mitochondria and chloroplast, but remember, in eukaryotic cells, those that are inside the mitochondria and chloroplast are of the 70S variety. Polyribosomes are actually short chains of ribosomes that are actually just stuck together. Eukaryotic ribosomes are similar to bacterial ribosomes in the fact that they do have a large and small subunit of ribonuclear protein or RNA. The full-size ribosome is 80S, and it's a combination of the 60S and 40S subunits that will come together to form the fully functional ribosome. So in this case, when the two subunits get together, they actually lose 20 pounds instead of the 10. And this just shows a conceptual drawing of the large subunit, which is the 60S, and the small subunit of the 40S coming together to form that ribosome. And we'll talk about again how ribosomes actually function a little bit later on to produce the proteins. The cytoskeleton, this is going to function in anchoring organelles within the cytoplasm, moving RNA and vesicles throughout the cytoplasm, permitting shape changes and movement of the cytoplasm. And there are three main types of protein that make up the cytoskeleton. Actin filaments, or what we refer to as the microfilaments, and these are the thin, small filaments, intermediate filaments, and microtubules that are made up of a larger protein and so are actually the largest of all of these filaments. And this is a fluorescent stained cell. This is the cell membrane in the red, and all of this green is the cytoskeleton. And so literally this gives the shape to the cell as well as performing other functions. So if we compare, again, the major elements of life and their primary characteristics, and you can go down and read through the chart, but you'll see that there are a number of differences and a number of similarities. Do they all contain nucleic acids? Absolutely. And nucleic acids, I'm talking DNA and RNA. Do they contain chromosomes? The prokaryotic and eukaryotic cells do, but viruses do not contain chromosomes. Okay. Is there a true nucleus? Only in eukaryotic cells. That's because they are the only ones that have the nuclear envelope or nuclear membrane. Eukaryotic cells reproduce by mitosis. They also produce sex cells, whereas the bacterial and viral cells primarily do not, even though there are a few of archaeal cells that may actually do sexual reproduction. Can they reproduce by binary fission? Although the prokaryotic cells do, eukaryotic cells, some can do that, while viruses can't. So certainly you can read on down through this. We're going to talk about a lot of these characteristics as we go through each of these. So true or false, eukaryotic chromosomes are visible during all portions of the cell life cycle. The what type of endoplasmic reticulum is responsible for packaging and transport of proteins? And what are vesicles that contain digestive enzymes within the cell? And true or false, mitochondria and chloroplasts contain 70 S ribosomes and their own circular DNA. So let's talk about the kingdom fungi first. Over 100,000 species of fungi have been divided into two primary groups. The macroscopic fungi, which includes all of the mushrooms, puffballs, and gill fungi, and the microscopic fungi of the molds and yeasts. Molds are multicellular, while yeasts are unicellular. The yeast cell is a single cell. It's round to oval in shape. It uses asexual reproduction primarily by the process of budding. It has an unequal cytokinesis. Molds grow long thread-like cells known as hyphae, and they're found in filamentous fungi or molds. They actually then will form what's known as the mycelium. So the mycelium, or the body of the mold, is actually comprised of groups of these hyphae that have grown together. Some fungi, some molds, actually grow what we call septate hyphae, where each individual cell is separated from the other by a barrier called the septum. And others, the cells just simply flow together in these hyphae, and so these are non-septate hyphae. Pseudohyphae are actually yeasts, and this is where the cell buds and that cell buds and that cell buds, and so it forms a hyphae-like structure, but each cell is actually a yeast unto itself. And then some fungi are actually dimorphic. At certain temperatures, some fungi will be yeast-like, but at lower temperatures or higher temperatures, they may actually grow more like molds. So here's our yeast. So a single body with all of the characteristics of the eukaryotic cell, and this is actually then showing the bud. So they undergo mitosis basically, and simply then pinch off apart with the separate nucleus. And so down here you can actually see the little buds that are coming off right there. And this is what happens with a pseudohyphae. So this yeast buds and that bud buds before it separates from the parent cell, and then that bud buds, and etc. And so we kind of grow these long chains of these budding cells that are still interconnected with each other. And so this is the pseudohyphae. The molds will actually grow from the mycelium, and this is our good old friend, a rhizopus mold or bread mold. This mold I don't like. I am highly allergic to this particular mold. And what you're seeing right here are what we call the aerial hyphae or the reproductive hyphae. And each one of these little black dots is actually going to contain the reproductive spores of this particular mold. Now, if the mold can acquire nutrients from a wide variety of substrate, we call this fungus a heterotroph. However, if the mold obtains its food source from dead plants or animals or dead organic matter, we call them a saprophobe or a saprophytic fungi. If, however, they must live on the body of living organisms in order to obtain their nutrients, we simply call them parasitic fungi. So this actually is a saprophytic mold that's growing here on some raspberries. And probably if I looked in the back of my refrigerator somewhere, I probably have some sort of experiment that's going on like this one. But this one actually is a parasitic fungi, and this is actually athlete's foot. Most microscopic fungi grow in loose association or colonies. The molds actually grow that mycelium, and this is this woven intertwining mass of the hyphae that make up the body or the colony of the mold. And again, some will actually form septate hyphae. The hyphae will be divided into segments by cross walls or septa. And all fungi reproduce asexually, and they reproduce by forming reproductive spores. So off of the mycelium, we will actually see reproductive hyphae that will produce then the reproductive spores. The rest of the hyphae are what's known as vegetative hyphae, and these are what are absorbing the nutrients from their environment, either from the dead organic matter or from a living organism. Fungi have many complex and successful reproductive strategies. As I said, all fungi reproduce asexually. However, some actually can reproduce sexually as well. So we have simple outward growth of the existing reproductive hyphae that produce the spores. In a few fungi, we actually have a process known as fragmentation. And this is basically a separated piece of the mycelium can now generate a whole new colony and start growing on its own. But the primary reproductive mode is by either an asexual spore formation or sexual spore formation. Fungal spores should not be confused with the resistant, non-reproductive bacterial endospores. Just keep that in mind. So there are several types of asexual spores that are formed, but we're going to look at just a few of them, of the more common ones. And the sporangiospores, these are formed by successive cleavage or binary fission within a sporangium, which is a sac-like structure that sits at the end of one of these reproductive hyphae. The kinidiospores, or conidia, actually are produced at the end of the reproductive spores without being enclosed by a spore-bearing sac. They kind of look like a dandelion head. And then some actually reproduce at the end of the reproductive hyphae by the production of what's called the macroconidia or microconidia, and I'll show you a picture of those. So here we have the sporangiospore. So we simply will have these cells that undergo binary fission within the sporangium. And then this capsule will actually rupture and release all of the reproductive spores all at the same time. Versus the kinidiospore, which will just simply then branch off from each of the ends of the reproductive hyphae and produce the dandelion-like head of spores. And these can just simply continually break free into the environment. The macro and microconidia actually will branch off of the reproductive spores and will form, usually in the macroconidia, a multi-septated structure that will hold spores, or simply the small spore itself. So what is this? Well, we see all the spores contained within the sac-like structure. I call these the lollipops. So these are sporangiospores. Versus this, that again kind of looks like the dandelion head up there. So this is a conidiospore. And here are the macroconidiospore and the microconidiospores. Now, sexual reproduction does occur in some of the molds. And what is the importance of sexual reproduction? Well, it's basically genetic diversity. Slight variation in form and function are potentially advantageous to the adaptation and survival of the species. So those that can actually undergo sexual reproduction adds to the genetic diversity of the organism. And what we'll see is that during sexual reproduction, again, all molds reproduce asexually. So they'll undergo mitosis and produce the asexual spores. But if two types get together, and we simply call them a plus and minus mating type, they can actually then form a cell by nuclear fission, nuclear fusion, to form a diploid cell. And this then is going to be our sexual reproductive spore. That will then, once released, undergo meiosis and go back to haploid numbers to produce haploid spores that will then begin the asexual reproduction over again, but now we have a new genetic combination. So three main types of sexual spore formation that we find in molds. This one's the easiest to recognize and is known as a zygospore. This is going to be one of the mating pairs and this is the other one. And they came together to form this diploid spore that will then be released and undergo meiosis in order to form the asexual spores again. This is another sexual spore that's formed on primarily mushrooms, but this is known as a basidiospore. So as the aerial hyphae grow up in the fruiting body of the mushroom, they actually will form these sexual spores then on the tips of these hyphae that have grown together and form these basidiospores that then are released into the environment. And then the last type of sexual spore is the ascospore. And if you can kind of see, all of these spores are lined up in this elongated sac structure known as an ascus. And again, the ascospore and the basidiospore are primarily found in molds that produce the mushroom. So how do we identify fungi? Well, we can isolate on specialized growth media that will grow fungi, but oftentimes fungi grow very, very slowly. We can observe microscopically and macroscopically from scrapings or other collected specimens. We can see if there are sexual spores that are difficult to detect, but if we see them, we oftentimes then are able to speciate based upon what sexual spore is actually being produced. And we can actually even use asexual spore-forming structures to help identify the genus and species. For example, penicillin only produces kinidiospores. So we can say that the penicillium mold, if we see a kinidiospore and we're thinking it's penicillium mold, then we probably have our identification. We can also look at the hyphal type, the colony texture, pigmentation, the physiological characteristics of the mycelium, and finally, we can even look at the genetic makeup. So, medical conditions that are caused by fungi that we're concerned about. Most human fungal infections occur through accidental contact with soil, dust, or some sort of carrier. The pathogenic fungi usually cause infection through accidental contact, but primary fungi can actually sicken healthy persons. Opportunistic fungi often attack people who are already weakened in some way. We actually have normal fungi that's part of the normal microbiome, but if something alters the normal microbiome or we have a weakened immune system, these fungi then become opportunistic and overgrow. Other medical conditions that can be caused by fungi are certainly allergies. I know, mold allergies, it's lovely, trust me. And neurological conditions due to fungal toxin production. So this is just some of the major fungal infections of humans. The superficial fungi either infect the outer epidermis or the epidermis, hair, even the dermis, and can go as deep as the mucous membrane, skin, or nails. Most of these are caused by Mycosporum, Trichophyton, Epidermitophyton. Candida albicans is a very common yeast infection seen in mucous membranes, and the Malassezia furfur is one of the ones that we now see a lot in the outer epidermis. So most of the ones that infect the epidermis, hair, or dermis are called dermatophytes, and they're also known as tinea infection, or sometimes called in general ringworm. Systemic fungal infections primarily affect the lung. They are inhaled, the spores are inhaled, and then can go from the lungs to many parts of the body. And we have several that we see. Histoplasmosis, caused by Histoplasma capsulatum. Blastomycosis, caused by the blastomyces yeast. And both of these, blastomycosis and histoplasmosis, are found in the Mississippi, Ohio River Valley Basin, and that does include southwest Missouri. Coccidioides immitis, causing coccidiomycosis or San Joaquin Valley fever, is primarily seen in the southwestern United States because it likes to be in dry, arid soil. And Cryptococcus neoformans, causing cryptococcosis, is actually a fairly urban disease because this is primarily found in dried pigeon feces. And there is one other called paracoccidiomycosis, called South American blastomyces, caused by Paracoccidioides brasiliensis, but because this is primarily seen in South America, we won't discuss this particular one in lecture. Other impacts of fungi, certainly they can have a negative impact on industrial production, since some of these are pathogenic to corn and other grains like wheat, soybeans. It's estimated that 40% of the yearly fruit crop is actually consumed by fungi. It, however, can have a positive industrial impact. Certainly, fungi play an essential role in decomposing organic matter and returning essential minerals back to the soil or the environment. They can form stable association with plant roots that increase water and nutrient absorptions for the plant. And many of our antibiotics are actually produced by fungi. Also, the production of alcohol, organic acids, and other vitamins actually are provided by fungi. Food flavoring. I mean, how many of us had mushrooms on our pizza? And the staples of life, beer, bread, wine. All of these are produced by fungi. So, blank is a type of fungi that derives nutrients from dead or decaying material. True or false? Fungal spores have a tough outer coating that allows them to survive extreme conditions. Blank are asexual spores that form and are contained within a sac-like structure. And blank are sexual spores that are contained within a sac-like elongated structure. And true or false? Pathogenic fungi require a host to complete their life cycle. Now we'll turn our attention to the protists or the protozoa. Algae and protozoa have been traditionally combined in the kingdom Protista, or we now call it protozoans. Subkingdom is algae, which we really will not discuss because, again, mostly it does not cause disease other than allergy or other types of things like that. And the subkingdom protozoa. So we'll spend our times with the protozoans. Protist is any eukaryotic, unicellular, or colonial organism that lacks true tissues. All of the protists are single-celled. The algae can form some colony-like structures, but still are simple cells unto themselves. So the algae are photosynthetic protists, and the big thing that I can say about them is these are the primary oxygen producers in the atmosphere. Seaweed and kelp are the most common recognizable algae because they are actually macroscopic. The algae exhibit all of the eukaryotic organelles, but they also have chloroplasts with green chlorophyll, and they can have other pigments that create yellow, red, and brown colorations. So just some examples of the algae, and I will say that this particular picture are diatoms, which are unique to each body of water, both fresh and salt, and these are actually microscopic algae which really form the basis of the food chain. There's nice pond scum that we can find around this area. And then this is a algae called Volvax that is a colonial type of algae. Algae are found worldwide in both fresh and salt water. Plankton is actually a floating community of microscopic organisms that, along with diatoms, forms the basis of the food chain. So they play an essential role in the aquatic food web and plays a big role in the entire food chain itself. The algae again produce most of Earth's oxygen within its atmosphere. However, there is a particular primary medical threat from a particular algae that's through injections of toxins during these red tides caused by these algae. The protozoans include about 65 or so hundred different species. Most members are harmless and are inhabitants of either water or soil. A few species are parasitic and are responsible for millions of infections in humans every year. Protozoans are single cell, containing again all the major eukaryotic organelles except for chloroplasts. The organelles can be highly specialized into structures analogous to actually mouths, digestive systems, very primitive, reproductive tracts, and actually legs or means of locomotion. Heterotrophic protozoa require their food in complex organic forms. And so this is what allows the protozoans to actually be found in so many different environments, is the fact that they can actually use a variety of different things for their food. Free-living species tend to scavenge dead plant or animal debris, so organic matter, and they help to actually get rid of the organic matter waste. They can graze on live cells of bacteria and algae, but there are few parasitic species of protozoan, and these live on the fluid of the host, such as plasmid or digestive juices. They may actually actively feed on tissues as well. So styles of locomotion. Some protozoan use what we call pseudopods or pseudopodia, and literally it means false foot. These can serve as feeding structures and actually form endocytic vacuoles that bring in food into the cytoplasm. Flagella, and these vary in number from one to several and can be located in various areas around the cell. And then cilia, and these typically are distributed over the entire surface of the cell for most of these protozoan parasites. So this is an amoeba, and this is actually the pseudopod. These pseudopods are formed by cytoplasmic streaming, and so the cytoplasm will simply move in the direction of the pseudopod, and then everything flows in that direction. This actually shows the flagellum on this particular organism, and this one also has something called an undulating membrane, which is actually a fold of the cell membrane that can do just that, undulate and assist the movement of the flagellum. And then this organism has all the cilia located all over it, and again, because of the recovery stroke, we see movement in one direction. All of the protozoas exist in the trophozoite stage at some point. This is the motile and feeding stage. This requires ample food and moisture in the environment for the trophozoite to remain active. And the other thing, this is the reproductive stage of the organism. Now, some of the protozoans only exist in the trophozoite stage, but some can actually form what's called the cyst. And this is a dormant resting stage with minimal metabolism going on. And these are formed usually when conditions become unfavorable for growth and feeding. And so there's no reproduction. However, cysts are a very important factor in the spread of disease. Many of these protozoan parasites can encyst in tissues that are then ingested by other animals, and the cyst then will, quote, hatch out and form a trophozoite in another host. So here's the life cycle. The trophozoite actively feeding and reproducing, but now the environment becomes dry. Cells begin to round up, they lose their motility, and we actually form a wall around the organism. So this is a mature cyst, and this can actually be encysted into tissues. But when moisture returns, the cyst wall breaks open, and this then reactivates into the trophocyte stage. The life cycles of protozoans vary from very simple to extremely complex. The best thing that I can tell you, if you want to know the life cycle of a particular protozoan, go to cdc.gov, type in the name of the protozoan, and find their life cycles. Some protozoan groups, again, only exist in the trophocyte stage. They never form a cyst. But many types will alternate between the trophocyte and the cyst stage, depending upon the habitat and environment. So the life cycle dictates the modes of transmission. Protozoan reproduction is primarily asexual through mitosis. They can, however, undergo multiple fissions, and each part then grow a complete new organism. And a few actually have sexual reproduction that will occur. Conjunction is the form of genetic exchange between two cells, and protozoans, similar to bacteria, can. So we can classify certain protozoa based upon how they use their flagella to move. Some have a flagella alone, or a single flagellum, or some have a flagella and amoeboid motion together, or some have a flagella and an undulating membrane. They certainly all have the single nucleus because they are eukaryotic cells. Most protozoans that reproduce sexually reproduce by the process called syngamy, where basically two individuals fuse together and then will undergo meiosis to form genetically diverse organisms. And again, some will form cysts, while others will always be free living in the environment in the trophocyte stage only. Those using amoeboid motion to move are primarily the amoeba. They use pseudopods for locomotion only, and they reproduce asexually primarily by binary fission. Most are uninucleate, meaning they only have one nucleus, and most of them are able to form a cyst. Most are free living, especially in water environments, and are typically not infectious, but there are a few that are infectious. Those using cilia to move, some have cilia in tufts for feeding and attachment. Most of them can develop cysts, and they may have macronuclei and micronuclei, meaning they may have one very large nucleus and then some smaller ones that also contain some DNA. They primarily reproduce by what's known as transverse fission. Typically, those that have cilia are elongated, and so they simply split in half along their long axis. Most have a definitive mouth and feeding organelle that's really kind of a primitive digestive system. Most of these ciliated protozoa are free-living and typically harmless. And then we do have a group that has no motility, and these are known as the sporozoa. Motility is absent in most of its representatives. They have very complex life cycles with well-developed both asexual and sexual stages, and the entire group is parasitic. Most protozoa can be identified to the level of the genus because of their unique appearance under the microscope. So what are some of the considerations when we are identifying these protozoa? Well, certainly the shape and size of the cell, the type and number and distribution of the locomotive structures. Do they have flagella? Do they move by pseudopodia? Do they move by cilia, or a combination. Whether they have specialized organelles or can they form cysts. The number of nuclei. Blood, sputum, cerebrospinal fluid, fecal or vaginal specimens smeared on a slide with or without staining. Oftentimes, if these organisms are present, we can actually identify them fairly easily. So, what are some of the important protozoan parasites? Well, first of all, let me define the term parasitology. Parasitology is the study of the protozoan and the helminths, or the worms and flukes. A parasite is a term used often to denote protozoan and helminth pathogens, even though truly a parasite is anything that takes from its host without giving anything back. But here are a few of the important protozoan pathogens. Of the amoeboid protozoans that move by pseudopodia, Entamoeba histolytica causes amoebiasis. This can occur in humans, but also in animals. The primary source is fecal contaminated water and food. The entamoeba is shed in the stool and picked up orally, so that we call this the fecal oral route. This causes usually a severe watery diarrhea, and people can actually die from the severe dehydration that Entamoeba histolytica can cause. Naegleria fowleri and Acantha amoeba, these are the two brain-eating amoeba. They are both free-living in water and usually cause no problems unless you snort water up your nose that has these organisms in them, and then they can actually go through the cribriform plate and get into the brain, where they then cause damage. Of the ciliated protozoan, Balantium coli is the only one that causes disease, and it actually causes disease primarily in animals. So I'm actually not going to talk about it. Of the flagellated protozoans, one of the big ones is Giardia lamblia. This causes giardiasis, also referred to sometimes as Montezuma's revenge or traveler's diarrhea. The reservoirs for this are certainly animals, other humans, fecally contaminated water or food. This causes, again, a severe watery diarrhea that can last for several days and cause severe dehydration. Another flagellated protozoan is Trichomonas vaginalis. This causes trichomoniasis. This actually can or does occur in humans, and this causes a severe vaginitis in the female and can cause a urethritis in the male. And in fact, Trichomonas vaginalis is considered to be a sexually transmitted infection. Trypanosoma brucei and Trypanosoma krusei causes trypanosomiasis. Trypanosoma brucei causes sleeping sickness or African sleeping sickness, while Trypanosoma krusei causes Chagas's disease. These both are vector-borne by insects. And then Leishmania, and there's several species, Donovani, Tropica, and Burlescentia. These causes leishmaniasis, and this is actually skin lesions, or can actually become more widespread and cause systemic disease in internal organs. This also is insect vector borne and primarily comes from animals. And then of the non-motile protozoan parasite, one of the big ones is Plasmodium, and this includes Plasmodium vivax, falciparum, and malaria, and this causes malaria. This actually is a protozoan parasite that infects the red blood cells that causes severe anemia. This is vector-borne by mosquitoes. Toxoplasma gondii. This particular protozoan parasite causing toxoplasmosis typically gives kind of a flu-like illness that most people then get rid of pretty quickly. The primary reservoir is animals for this, and primarily cats. It can be vector-borne by a few mites, but that's kind of unusual for toxoplasma. Most people that are infected with Toxoplasma gondii either got it from an infected cat handling the feces, or they can insist in muscle tissue, and so by eating undercooked meat. Primarily, most people that get toxoplasmosis don't even know they have it. They shed the parasite out of the digestive tract very quickly, and so they usually feel better in 2 or 3 days. However, Toxoplasma gondii can cause severe problems with the developing fetus, and it can cross the placental barrier from the maternal bloodstream into the fetal bloodstream, where it can cause a number of defects. Cryptosporidium parvum, this is a very small protozoan parasite that lives in water. And so again, primarily fecally contaminated water or food is going to be where we find this. Causes cryptosporidiosis, causes severe diarrhea with cramping, and certainly again severe dehydration. And then cyclospora, this causes cyclosporiasis, and again, intestinal symptoms are the primary thing that we see. And this can be contaminated water or fresh produce that is produced using fecal material for fertilization. So let's look at a few of these. Trypanosoma brucei causing African sleeping sickness, and Trypanosoma krusei causing Chagas's disease. Both of these organisms are long crescent-shaped cells with a single flagellum, and what they do is they are in the bloodstream where they cause the destruction of red blood cells. So here's a couple of these protozoan parasites in the bloodstream. Both are transmitted by blood-sucking vectors. Trypanosoma krusei is actually vectored by a group of the trematode bugs. This is actually a large blood-sucking bug, oftentimes called the kissing bug in this region of the country because it likes to bite in and around the face. And Trypanosoma brucei is transmitted by the tsetse fly, which is a fly found only primarily in sub-Saharan Africa. In Chagas's disease that we see in the United States, the trematome bug actually will take a blood meal, and the parasite then will actually swarm into the bite and get into the bloodstream, where they then will cause damage to the red blood cells as well as several other cells. And then they actually can encyst, and they multiply by binary fission. And then as they float around in the trophocyte stage still, another trematome bug will pick up the blood meal, and then here we go again. And yes, we do have these bugs in this area. Now there is a bug called the wheel bug that is very similar in appearance to the trematome bugs in this area, but it's very hard to actually distinguish. So I just stay away from all the large, ugly looking bugs. Entamoeba histolytica, causing amoebic dysentery. This is the fourth most common protozoan infection found worldwide. The disease is transmitted through fecally contaminated food or water that has the entamoeba that was from someone else who had it. So true or false, algae don't cause any type of human disease. Describe each style of protozoan locomotion. All protozoa have a trophocyte form, but not all produce cysts. And what is the term that is most often used to describe protozoan and helminth pathogens? Now, let's look at the parasitic helminths. Helminths include the tapeworms, the flukes, and the roundworms. The adult stages are usually large enough to be seen with the naked eye. They can range in size anywhere from about 1 millimeter to 25 meters in length. The flatworms primarily are going to consist of the tapeworm. The tapeworm is kind of a unique worm in the fact that, one, it actually has a head structure that has what's known as the scolex. The scolex contains suckers and a ring of crown, or a crown of thorns, you might say. And this is not really shown well in this picture. I'll show you another one in a moment. But what this thing does is it hooks onto the side of the intestinal tract and then sucks up nutrients from the nutrients within the intestinal tract itself. Then, as it grows in length, each one of these little segments, called the proglottids, is actually a reproductive packet. Tapeworms are true hermaphrodites, meaning that they have both ovaries and testes within each one of these proglottids. Now, they can reproduce with another flatworm, but they can actually reproduce on their own. And as these proglottids mature and are reproduced, they then towards the end become nothing more than large packets of tapeworm eggs. And these proglottids then break off and are shed out with the feces. The flukes are primarily a large flat structure like this guy. They have an oral sucker, they have a primitive digestive system, and again, they are true hermaphrodites containing both ovaries and testes. Again, they typically do tend to reproduce with another organism, but they can reproduce by themselves. The roundworms include several species, and by their name, they are round. These typically have two species, males and females, and this is the female Ascaris lumbricoides, the common roundworm, and this is the male. This is actually the egg, and again, why we study some of these helminths in microbiology is because the eggs and some of the larvae stages are actually microscopic. So, generally, the worms and flukes are multicellular animals equipped with primitive organs and organ systems. The reproductive tract is the most developed in these organisms. They have thick cuticles on the outside for protection, and they have mouth glands for breaking down the host's tissues so that they can get to their nutrients. Or like the tapeworms, actually just take the nutrients from the host's nutrient stream. Complete life cycle includes the fertilized egg, larvae stage or stages, and the adult stage, which is the reproductive stage. The majority of the helminths derive nutrients and reproduce sexually in the definitive host's body. Nematodes, the sexes have different morphologies. They're what we call dioecious. They have both males and females, and this would include the roundworms. Trematodes, the sexes are separate or they're hermaphroditic. And hermaphroditic meaning monoecious, meaning they have both sexes in one organism. So the male and female sex organs are in the same organism. And cestodes are generally hermaphroditic. Transmission of the egg or larvae to the body of another host, either through a different or same species, starts the general life cycle of most of these helminths. The intermediate or secondary host may also be included, and this is the host in which oftentimes the larvae develops until it can reach to the primary or definitive host. The definitive host is the host that houses the adult cycle, and it's where reproduction will occur. Sources of human infection for most of the helminths is going to be food, soil, water, or infected animals or other humans, and primarily most of these are going to be through the fecal-oral transmission. So oral intake or penetration of unbroken skin by fecally contaminated water soil. Humans are the definitive host for many of these helminths and the sole biological reservoir for about half of these helminths. Animals or insect vectors can also serve as reservoir. We share in common as many of the roundworms with our domesticated animals. In fact, when I see a puppy or kitten, the first time I just deworm it immediately for roundworms because I know oftentimes there's children in the home and they can pick up fecally contaminated things and put them in their mouth, and we might get a roundworm started in the child. So here's just some of these worms. So the nematodes, the roundworms. Primarily intestinal nematodes include the Ascaris group, the pinworms, which is Interamoebus vermicularis, and I'll talk a little bit about this, and Trichinella spiralis. Of the flatworms, schistosoma or the blood fluke, this is primarily spread to humans by ingestion of fresh water containing the larvae stage or eating snails. And Teniasolium and Diphyllobothrum lathum, these are the pork tapeworm and fish tapeworm respectively, and these two actually can infect humans as well. We also can see the beef tapeworm that can infect humans, and there is one that is only found in humans. So let's just take a look at one of these, the Interamoebus vermicularis. This causes a very common infestation of the large intestine, and I use the term infestation because this really truly doesn't usually cause an infection. It's just usually more of an irritation. Worms range from about 2 to 12 millimeters long. They're tapered and cylindrical in shape. They have a simple, uncomplicated infection that usually does not spread beyond the intestinal tract. And these are kind of unique in that the female actually will, when they're ready to lay their eggs, migrate down to the anal opening. This means that the eggs then will be associated here in the skin of the anus, and with the next fecal deposit, the eggs will then be passed out of the host. So a lot of times what we can do to make a diagnosis of pinworms in children especially, because as you might think, these little worms crawling around in your anus causes you to itch. So if you look at children oftentimes, what you'll find is that they can be scratching their butt, and oftentimes these females like to lay their eggs when it's kind of quiet and when the child is still, so sleeping. And so your butt itches. What do? You're going to scratch it, and then you're going to reinfest yourself with the egg again. However, it can also come from another individual. So how can we identify these worms? Well, one, we can look at their shape. Are they flat? Are they round? What about their size? Absolutely. We can look at the degree of the development of organ and organ systems. We can look to see if they have the presence of hooks, suckers, or other specialized structures. What's their mode of reproduction? Are they dioecious or monoecious? And what are their kinds of hosts? Both intermediate or secondary hosts, and what is the definitive hosts of each? What's the appearance of eggs and the larvae? So microscopic detection of the adult worm or the larvae or eggs is one of the most common things that we use. And oftentimes all of these have distinct shapes or external structures or internal structures that make identification very easy. Normally I diagnose roundworms, hookworms, pinworms, even though I don't see a lot of pinworms because dogs and cats can't get them, but some of the other roundworms as well, I actually see the eggs in a stool sample. Occasionally we culture these to verify all life stages. So here's just a variety of helminth eggs. So this is Ascaris lumbricoides. Okay, this is actually a tapeworm egg, and I can see that because if you'll notice, it's really hard to do with the pointer, but there's 4 little lines in there, and those are actually what will become the hooks of the tapeworm. This is actually a hookworm egg packet. And this is actually a whipworm egg. And then these other ones are various other ones that we see. There's about 50 or so species of helminths that parasitize humans. They typically are geographically restricted, so some only are seen in tropical regions. But yearly estimates of worldwide cases are typically in the billions. Conservational estimates that there are 50 million or so infestations of parasitic worms in the U.S. alone every year. Humans evolved in the constant presence of these helminths. Only recently have humans evolved into a, quote, helminth-free existence. Absence of helminth infections, though, may actually be a detriment in the fact that it may contribute to autoimmunity and to certain allergies. I'm not saying, however, you need to go out and get a helmet. Don't misunderstand me. So, as we look at the variations among the eukaryotes, the level of complexity, again, protozoans will always be unicellular. Fungi can be unicellular or multicellular. Same thing with algae. The helminths are going to be multicellular in the adults, but unicellular in their ova or larvae. Cell walls, protozoans don't have one. The fungi can either have cell walls made up of chitin or cellulose. Algae, it's primarily cellulose. And the helminths do not have cell walls because they're typically multicellular. Their cytoplasm, what's their nutritional type, their motility, and some important structures that they each could possibly form. So take a look at this chart just to compare the variations. So last concept chat. Describe the differences between nematodes, cestodes, and trematodes. In helminths, what is the most developed organ system that is primarily seen? Some helminths are what? Having both male and female. And then what are the ones called that have only a single sex or have both sex organs in one organism? True or false? Some helminths produce millions of eggs to overcome a high mortality rate of ova. Okay, so join me for the next chapter and see you later.